Howdy, this is Lab Report 5, IMViC tests, Indo test, methyl red, Voges, Proskauer, MRVP broth, and citrate test. Group 2, Ana Martinez and
And Garcia, Microbiology Fall 2024.
Introduction and Significance. The purpose of this lab is to perform the IMViC tests, Indo test, methyl red, Voges Proskauer, MRVP broth, and citrate test, mainly to distinguish Escherichia coli from Enterobacter, cloacae, and other related species. These are biochemical tests for identification and differentiation of bacteria. The materials and methods for the indole test, tryptophan broth. These are the materials. Separately inoculate the bacteria listed aseptically into the labeled tryptophan broth tubes using an inoculating loop. Incubate for 24 to 48 hours, then add 0.5 milliliters of Kovach's reagent. Observe the tubes for any color changes. The background information. The indole test determines if the bacteria can produce indole from the amino acid tryptophan. If the bacteria have the enzyme tryptophanase, then it can break down tryptophan into indole, pyruvate, and ammonia. Kovach's reagent reacts with indole to produce a bright red compound at the top, which means it's indole positive. If the bacteria does not produce a red compound at the top, then it is indole negative. The materials and methods for the methyl red and Voges Proskauer MVVP broth. These are the materials. Separately, inoculate the bacteria listed aseptically into the labeled MV or VP broth tubes using an inoculating loop. Should be a total of 6 tubes. Incubate tubes for 24 to 48 hours, then add 1 to 2 drops of methyl red to the MV tubes and add 15 drops of reagent A and 5 drops of reagent B to the VP tubes. Color changes should be observed after 30 minutes. The background information. The methyl red test determines if the bacteria can perform mixed acid fermentation of glucose, resulting in the production of stable, strong acids. A positive result would turn the test tube red, meaning the bacteria produce stable acids and lower the pH of the medium. The Voges-Proskauer test determines if the bacteria can produce neutral end products such as acetone and 2,3-butanediol from glucose fermentation. A positive result will turn the test tube red, butanediol, meaning the bacteria performed butanediol fermentation. The materials and methods for the citrate test. Separately, these are the materials. Separately, inoculate the bacteria listed aseptically into the labeled citrate tubes using an inoculating loop. Incubate and observe any growth or color changes. The citrate test determines if the bacteria can utilize citrate as its sole source of carbon for metabolism and growth. Bacteria that produce the enzyme citrate permis can transport citrate into the cell, where it gets broken down into oxaloacetate and further metabolized, releasing carbon dioxide. A positive result would turn the test tube media blue, indicating production of alkaline products. A negative result will have no bacterial growth as well as no color change. The results and observations. The following slides are the results and our observations of the incubated bacteria in the test tube media. The pictures are our results and the observations are based on what we recorded on our lab notebooks.
Results and observations, indole test, tryptophan broth. For E. coli, the top red layer was positive with hazy movement, which means the bacteria are motile, and bacteria contains the enzyme tryptophase. For Enterobacter cloacae, there was a yellow top layer followed by a green layer indicating a negative test. It was also clear with no movement, indicating that the bacteria are non-motile. Therefore, the bacteria does not contain the enzyme tryptophan. For unknown tool 2, the yellow top layer indicated a negative result, but there was also hazy movement indicating that the bacteria are motile. Therefore, the bacteria does not contain the enzyme tryptophanase. For methyl red envy broth, Escherichia coli was red at the top, indicating that it was MR positive, as well as hazy. Mixed acid fermentation that produced stable acid end products that lowered the pH. For Enterobacter cloacae, it was also red at the top, followed by an orangish layer, indicating that it was MR weak positive. It was also clear and produced some stable acid end products that lowered the pH. In reality, however, it should have turned orange-yellow, negative. It should have undergone butanediol fermentation and produced neutral end products. For unknown 202, it was also red at the top, indicating that it was MR positive and hazy. It therefore displayed mixed acid fermentation that produced stable acid end products that lowered the pH. Bulges Proskauer VP broth. For Escherichia coli, it had a whitish top layer indicating that was negative and hazy. Mixed acid fermentation that produced stable acid end products that lowered the pH. For Enterobacter cloacae, it also had a yellowish brown top layer indicating that it was a negative reaction and hazy as well. It should have turned red positive. It should have undergone butanediol fermentation and produced neutral end products. And for unknown 202, it had a white top layer indicating that it was a negative reaction and hazy. Mixed acid fermentation that produced stable acid end products that lower the pH. Finally, for the citrate test, Simmons citrate agar, Escherichia coli experienced no growth and no change in color, retaining its original green color, which indicating that was a negative result. Citrate was not utilized, therefore the enzyme citrate permease was not produced. For Enterobacter cloacae, there was some growth and orange stain indicating that was weak positive. There was also partial citrate utilization, possibly that was not incubated long enough to turn blue, but it should have turned blue, indicating a positive reaction. It can actually use citrate as its sole carbon source by producing the enzyme citrate permease. For unknown 202, there was minimal growth but no change in color, retaining its original green color, indicating a negative result. There was also a yellow stain at the bottom, which might have been the result of possible contamination. Citrate was not utilized, therefore the enzyme citrate permease was not produced. In conclusion, these tests are helpful in identifying and differentiating bacteria based on their metabolic capabilities. Based on what metabolic capabilities a bacteria has, it determines whether they can grow and thrive in an environment with specific conditions. Thank you.